Hello students, now we will discuss about some of the points where the G1 phase, S phase, G2 phase of interphase during cell cycle and cell division are checking whether that stage is completely or successfully completed or not. Those points are called as checkpoints. So we can write like a checkpoint is a stage in eukaryotic cell cycle at which cell examines internal and external conditions and cell decides to move to the next stage or not. We can also say checkpoints are controlling mechanisms. In eukaryotic cell cycle. Now, how many checkpoints are there? There are totally 3 checkpoints. In which time means stage means interphase. We know that interphase is the time period when cell is preparing itself for either mitotic division or meiotic division. During this time, cell prepares itself for next M phase, which includes mitosis or meiosis. First one, we have said 3 checkpoints. First one is G1 S checkpoint, means between. So if this is the interphase time period, I have drawn circle, it doesn't mean that it is rotating like this, just I am indicating as time period. So sometime we are giving for G1 phase, sometime for S phase, sometime for G2 phase, and here will be M phase, and here will be cytokinesis, later will be G0 phase. So this line indicates the time period of completion of G1. So here by this time, the cell checks whether that G1 phase is successfully completed or not. Second one, G2 M phase. Third one, spindle fibers formation at anaphase to check. This can also be written as at the end of metaphase. First one, G1 checkpoint. At G1 checkpoint, there are some other terms. In eukaryotic cell, under eukaryotes also, in mammalian cells, this G1 checkpoint, it is called as restriction checkpoint, and in yeast cells, this G1 checkpoint is called as starting checkpoint. or start point. Here, in this G1 checkpoint, first or initially, DNA damages are checked, if there are any damages or not. DNA is checked for any damages. Next, secondly, synthesis of or synthesized enzymes, ATP, and proteins are checked. Next, thirdly, CDKs are checked. What are CDKs? Cyclin dependent kinases. These are used as initiators for transcription. When cell finds each and every means point or important substances are perfectly or completely formed or not, then only cell will be moving into next S phase. So second one, G2M phase. It checks the changes happened in S phase and synthesizes the reduced biomolecules like enzymes, proteins, etc. if necessary, and then moves to M phase. Next one, spindle fiber formation at M phase. Spindle fiber formation checkpoint. This is also called as M checkpoint. This happens at metaphase, anaphase transition. What does it mean? It means that it happens at the end of metaphase and at the beginning of anaphase, because during metaphase we can see all chromosomes are aligned on the equatorial plane. In the next stage, means in anaphase, here will be the centriole. The spindle fibers will be attached to the kinetochore of the chromosome. So what is kinetochore? If this is the chromosome, middle part will be centromere. Now, on either side will be, here will be the kinetochores. So here, the cell cycle will check whether spindle fibers are exactly attached to the kinetochores to drag the chromosomes to opposite side during anaphase or not. To drag the chromosomes to opposite poles during anaphase, at metaphase ending, cell will check whether the spindle fibers that are formed from the centrioles are attached to the kinetochores properly or not. This phase is called M checkpoint or spindle fiber formation checkpoint.